Evening, good afternoon. Can everyone hear? Peter, can you make sure they can hear me? I don't want to deny anyone of my voice. That's just is my. Good afternoon and welcome to the University at Albany's East Campus, the home of our School of Public Health and the Cancer Research Center. I am Fardeen Sanai, Vice President for Development and Executive Director for University at Albany Foundation. I bring greetings from University at Albany Foundation President, Mr. George Hurst, and University President, Dr. Jones. Dr. Jones is traveling and just got to Albany, and we have to give George Hurst a pass. His daughter, his only daughter, just got into town, and he's going to spend some time with her. University at Albany Foundation owns and operates the East Campus through its affiliate, University at Albany Biodevelopment Corporation. It is fitting that we are in this conference center as Mr. Mazary, President of the UABDC, along with Mr. Dan Hogarty, led the efforts to purchase the former Sterling Winthrop Pharmaceutical Complex in February of 1996. It seems just like yesterday, correct? Since that purchase, the foundation has worked with the University at Albany to develop the 95-acre site using a collaborative model for a biotech campus which provides leased office space and high-tech laboratories with universal access to common spaces and services including laboratory support, state-of-the-art conferences and meeting facilities, and on-premise recreational and dining opportunities. The East Campus is intended an environment which is life science technologies can be developed into a commercially viable businesses, and also to foster the education and clustering of a workforce with the skills necessary to sustain such enterprises. The University at Albany conducts activities on East Campus, such as the School of Public Health, the Cancer Research Center, and the Student Loan Service Center, which are complemented by the operations of a variety of tenants and collectively represent a range of health-related efforts through the spectrum of education, investigation, research, development, and production design. This campus is a perfect example of how our benefactors and volunteers help us provide the margin of excellence in our mission of education, research, and service. Thank you to Dan Hogarty and Ellen Hogarty and their daughter, Sheila Mendelson, for creating the Hogarty Lecture. Because of your support through the Hogarty Family Foundation, this lecture series continues to educate us on a broad range of topics related to cancer. We had such a strong response for tonight's talk that we are simulcasting the lecture into Atrium. So thank you everyone, both here and at the Atrium. Important work is being done at our East Campus that will improve the health of our society. The School of Public Health, ranked 25th in the nation by US News and World Report, trains the next generation of public health professionals who will lead the fight against health disparities, obesity, substance abuse, social violence, environmental hazard, and more. Our Center for Excellence in Cancer Genomics is committed to research that will discover the genetics and environmental origins of cancer and lead to finding a cure for a disease. Our state-of-the-art research facilities have a strong ties to our School of Public Health and is affiliated with New York State Department of Health Wadsworth Research Laboratories. Critical work is underway here at the University at Albany's East Campus. I anticipate that we will be further enlightened by tonight's lecturer, Dr. Rosemary Rochford. To tell you a bit more about our keynote speaker, I would like to introduce Dr. Douglas Conklin, Associate Professor in the Department of Biomedical Sciences. Thank you again. Doug?
Thank you, Fardeen. So, and thanks to everyone for coming. This is a special night for the denizens of the Cancer Research Center. You've probably heard about the simulcast from everyone. This is an experiment. We've unfortunately got a little bit of a reputation for failed experiments in this building, but so please bear with us. For those of you in the other room, there's crackers and cheese. The next hour is likely to be binding as well as spell binding. We had entertainment out in the atrium. It was Professor Dave Hosley and a student who's in the music department. He's graduating. Brian O'Connor was on the bass. I'd like to personally thank them for taking the edge off the marble and glass in the other room. And there are Albany peeps, so it's good. And then I'd also like to I guess, get into a little bit of logistics very quickly. So if you're in the atrium and you've got a question for Dr. Rochford, try to sort of migrate this way toward the end of the talk so that you'll actually be able to catch her as she runs for the train. So I'd like to introduce tonight's speaker, Dr. Rosemary Rochford, who's Vice President for Research at Upstate Medical University in Syracuse. Rosemary's from Colorado and got her Ph.D. at UC Irvine. She's worked on creepy viruses most of her career. Is fascinated by them. And she after her postdoc, she postdoced briefly at Scripps Institute in La Jolla, and then went to work at another school of public health at the University of Michigan before then moving to a professor position at Upstate, where she's now a professor in microbiology and immunology, as well as being vice president for research. So she's very talented. She's going to tell us tonight about Burkitt's. I'm very interested in Burkitt's. My sister-in-law was diagnosed with Burkitt's, and so in my family, then everybody comes to me with lots of questions. She's okay now, but the most interesting question that I got, I said, my sister-in-law has been diagnosed with Burkitt's, and all the experts that I talked to, they wanted to know. They said, I didn't know your sister-in-law lived in Africa, which then I had to do a little digging, and it's a really fascinating story, and I'm looking forward to it. Dr. Rochford.
Well, see if I'm mic'd. Can you hear me? All right, good. So thank you for the invitation to come, for Martin and Doug for the invitation to be here, for the Hogarty family for supporting this lecture series. Cancer is an important area. It's nice to be able to have a public lecture about cancer research and be able to share with you what I've been doing in Africa. And I have this picture up here because when we think about going to Africa and you talk to people that you usually think about going on safari, seeing the big five, lions, elephants, rhinos, and so on. I've got my elephants here, and this is Mount Kilimanjaro in the distance. I think the snow's probably decreased a little bit since then. But I want to talk to you about a different reason to go to Africa, and it's to do cancer research there. So I study a children's cancer called Burkitt's lymphoma. This is the endemic form. Actually, as Doug stated, there's 3 different types, the endemic, the AIDS, and the sporadic. In Africa, we're looking at the endemic form there. It strikes children. These are all pictures of children with Burkitt's lymphoma. I took all of these photos there. And I always like to bring this picture in mind because when I first started to work on Burkitt's lymphoma, it was sort of an abstract concept about this cancer that was in Africa. But for me, going there, I always talk about being face-to-face, and it really brings home to me why it is that we do the work we do. Underlying these faces, though, is some very stark statistics. When we think about cancer, in 2012, 8.2 million people are estimated to have died of cancer. The majority of those deaths, however, occurred in the less developed world. And by 2030, this is a statistic that the World Health Organization has their attention, as well as the National Cancer Institute in the US. 7 out of every 10 new cancer cases will occur in the developing world, in a place where there's very little resources for treating or preventing cancer. So today what I'm going to do is talk about a little bit about a background on Africa. I call it by the numbers, just to give a framework for where I work. The challenges of cancer research in Africa from my own experiences and what I've dealt with, some of it anyhow, and then examples from my own research program that we have in Kenya. And I'm going to talk about some of our field studies on the etiology or the cause of Burkitt's lymphoma. I'm going to talk about some of the work we do in capacity building to train the next generation there to be able to do the work in country. And then some work we're doing to start with on cancer registry to know the burden of disease. So Africa by the numbers. I always like to remind people, when we talk about Africa, sometimes people think it's a country, but it's a continent. It's very diverse, 53 nations. The continent of US, Europe, and Asia would all fit within the continent of Africa. So just to give you a sense of how large it is, when you're flying across the Saharan desert going down to Kenya, it's a 4-hour flight. And if you think about going across this country, that's 4 hours of flying across desert. 14% of the world population is there. And the percentage under age of 25 years, 71% of the population is under 25 years of age. So when you talk about studying a children's cancer, and it's the most common children's cancer, it affects a lot of children within that population. The life expectancy in sub-Saharan Africa, the equatorial plane is right there in this region, is only 46 years. Very grim statistics. And sadly, the most common cause of death is as AIDS. So it's a continent worth paying attention to and thinking about, but it's a sort of a vast continent to understand. And when we try to think about doing cancer research in Africa, there's a number of issues that we have to deal with. The first one is thinking that awareness that cancer is an actual health problem in Africa. So many of the indigenous languages don't even have a word for cancer. A lot of people, when they develop cancer, the children, we've talked to people, actually it was in Malawi where they have these large jaw tumors, they think it's a curse. There's no available treatment in many rural areas, so it's just essentially a death sentence. But at the local level, there's very little awareness of what that cancer is. When we think about it at a national level, most of those countries in Africa, 53 nations, very few have a cancer control program. Very little government-mandated studies of cancer. That's beginning to change a little bit, but it's not good. And we think about it at an international level. We also have to think about a certain bias in research in that it used to be thought that infectious diseases were the problem of the developing world, or the third world as we used to call it, and chronic disease and cancer were the problems of the first world. That's not obviously true, and I like to think about Burkitt's lymphoma was described in the Western literature in 1958, but it's obviously been there for a long time. And I like to show this slide here. This is a picture, one of my students in Kenya found this one. It was in a museum, Western African face mask circa 1880s, and you can see this very Picasso sort of abstract art-like face. And you might think this is sort of a tribal depiction of sort of an abstract ideology. This, sadly, is a young boy with Burkitt's lymphoma that I took in 2007, and I suspect that this mask is actually a representation of a child with cancer. Another challenge is so we don't know an awareness of the disease, and then how do you begin to sort of think about what the burden of that disease is? How much cancer is actually in Africa? So how do we get and measure that burden of disease? So that highlights the role of cancer registries. When we think about this, in this country, we have a national mandate to know what the incidence of cancer is. We can do a hospital-based registry, and this is where we look at cancers that come into a certain clinic, and you can report that and record that. But the best registry, and what we do in this country, is a population-based registry. And that means you send people out to find all the cases of cancer that occur within a certain region. So you go to hospices, different doctors' clinics, different hospitals, a number of different cases to see if you can gather it in for the entire population. Those cancer cases, you get details of the type of cancer, the person that's affected, type of cancer, treatment, follow-up, especially with respect to survival. That's what we do in this country as best we can. And you get cases, again, from all around the world. This is a percent of population covered by cancer registries in a publication by the International Agency for Research in Cancer, IARC. It's an arm of the World Health Organization, and they publish this volume every 5 years. If you look at the continent of North America, about 83% of the population, they've captured data on the type of cancer that occurs within 83%. If you look at Africa, it's 1%, right? This is a really very low number. Not so good over here in Asia as well. And if you look at it and actually look at the countries where they gathered the data, it's actually, I think, just 4 or 5 countries. And if we think about this, this would be trying to understand the cancer incidence, say, in West Virginia, where there's a lot of cigarette smoking, compared to Southern California, where there's very little. So it really doesn't give us a sense of the true burden or scope of the disease within the continent of Africa. I'll skip that. So the other challenge is under-resourced clinical research infrastructure. This, you can't see so well. This is a pathology lab at the major national hospital, Kenyatta National Hospital in Nairobi, Kenya. This is the hospital that's the major referral hospital for the entire country. When I went to this pathology lab, there was tubes and jars of specimens that had come from all over the country that had been there for months and months and months. It typically takes about 6 months to get any diagnosis based on pathology. This just shows you, just in terms of under-resourced clinical infrastructure, this is a figure looking at the number of people served by a single radiotherapy center in Africa, and the countries that have greater than 30 million population where there's only one radiotherapy center for the entire population. So very little access to treatment, few specialized oncologists per country, and therefore lots of lack of treatment options. And this is true throughout most of the continent of Africa. And I just show these pictures as well because these are photos that I've taken from Kisumu, Kenya, when at the provincial hospital that's the major treatment hospital for children with Burkitt's lymphoma. And usually, typically in the wards there, you'll see multiple beds, and there's multiple kids per bed, and the mothers will come with them during the treatment period. They usually stay in the hospital for about 2 months or so because it's hard to get back and forth because they live in rural areas. And the mothers will sleep on the beds with the children at the hospital. This is a little boy sitting on a hard bench, and again, it's hard to see here. It comes up better. I'm getting his chemotherapy infusion. When we think about our cancer centers at Upstate Medical University, we're opening a brand new cancer center, which is lovely and wonderful, and these beautiful infusion rooms, as they should be, but this is much more the reality in many parts of Africa. And this is a child with advanced disease, and this is also fairly typical that they often don't come into the disease as much later. So when we think about trying to prevent or treat cancer, we always try to get that early detection, and treatment is much more successful. Very often the children come in. This isn't an abstract, rare observation. It's fairly common. Oftentimes the family will take them to traditional healers. You'll see scar marking on the face before they finally bring them into clinic. And then lastly, just the lack of training in cancer and cancer research. If a government doesn't think that there's a problem in cancer and you don't have awareness, you're not going to have cancer training programs at the medical schools. You don't have cancer, any kind of description. We have great molecular biology where kids learn about apoptosis and cell cycle and all the ways that cells can go bad. That's not available there. Most of the focus is really on infectious disease and training, so there's very little there. So there's a number of challenges in thinking about working in Africa. So you might say, what are you doing there? But what I'm going to tell you now is a little bit of a story of Dennis Burkitt, and one of the reasons why I think it's very important to work there, many reasons to work there. But I'm going to tell you a little bit of the history of this cancer that I study, and then go into some of the work. So Burkitt's lymphoma, it's a monoclonal B-cell lymphoma, so it's a cancer that comes from the B cells. The B cells are the ones that make the antibody, and as I said, there's 3 types. This endemic form that we find in equatorial Africa. You can also find it occurring in AIDS patients and sporadic, but what I'm going to talk about is focusing on the endemic form. It has this chromosomal translocation, and what this means is that this, what's called the c-MYC gene, it's an oncogene, gets put in place next to a gene that turns on the immunoglobulin, the thing that makes the B cells and makes the antibodies. And this c-MYC oncogene gets overexpressed, and that's what drives those cells to proliferate. And that's characteristic actually of all 3 types. It's a rapidly growing cancer. It's fatal if untreated. So it's considered to be a medical emergency, and typically if a child comes in with a fairly large jaw tumor, they'll start treating with chemotherapy before they even wait for any pathology. And oftentimes the diagnosis that it's not Burkitt's lymphoma is that it doesn't respond to treatment. But actually, when the children are treated, it responds fairly rapidly. So the story of Burkitt's lymphoma actually starts with Dennis Burkitt right here. And Dennis Burkitt was an Irish surgeon, which I always like to say he's Irish because I'm Irish, but he trained in England. And he was posted down to the Malago Hospital in Kampala, Kenya. So this was in the 1950s, and he was interested in the field of geographic medicine. And this is actually, that's a picture of me with Jackson Oram there, and then that's pretty much how it looked back in the 1950s. This is 2008. And what he saw was this young children with these jaw tumors, as I talked about. They're very striking. Dennis Burkitt had never seen anything like that in his training in England. And I always think about, I like to bring up this story because oftentimes right now in our field we talk about from bench to bedside. But although I'm not a physician, I always say it has to start with the bedside to begin with. And Dennis Burkitt is the classic example of a curious physician who took that curiosity further. And being a curious physician, he did what any good scientist would do. He wrote money for a grant, and he got a grant for $100, so the equivalent, big money. And he asked the question, and we wanted to see the distribution and understanding why children get Burkitt's lymphoma. So this is a map showing this case, and the black dots, what he did is he basically mimeographed a diagram of a child with Burkitt's lymphoma and mailed it out to clinics throughout Africa. And he got the responses back and he mapped them to where they came from, and you can see these dots all across Africa here. Now what's striking about that is it's a limited distribution. It's not everywhere in Africa. It's actually in the equator belt. And the pink areas here represent, the areas where we have malaria that's holoendemic malaria. Now, when I, excuse me. I got my Ph.D. in molecular biology and biochemistry, so we never knew what holoendemic malaria is. I've learned since, and so I'm going to tell you a little bit what that means, because we hear about malaria a lot as well in terms of work being done with that. So malaria, I'm just going to give you a little bit of background on malaria and get you to be excited about parasitology, since part of the talk is about parasites. It's transmitted by a mosquito called an Anopheles mosquito. It's a mosquito that bites at nighttime, and the mosquito transmits the parasite. It gets to the liver, replicates in the liver for a little bit, and then gets into the red blood cells, and it has this red blood cell stage for quite a while, and this is where you get these cyclic fevers. It's the red blood cell stage when those blood cells burst open in lice that causes the anemia and the cerebral malaria and the morbidity and mortality that we find associated with malaria. Ultimately, has a sexual stage that then gets picked up by another mosquito and then transmitted and continues the cycle again. Now, there's 5 different species of malaria that 5 different species of plasmodium that cause malaria in humans. The most severe one is caused by Plasmodium falciparum, and this is what we see in Africa. Places like India have a different type of malaria called Plasmodium vivax, but in Africa it's the Plasmodium falciparum. This is the number. It seems like they're going down a little bit since the introduction of bed nets, but it's been estimated that approximately 1 million deaths every year in young children are caused by P. falciparum malaria. The striking thing about this malaria, and this is the piece that took me a while to understand, is immunity to malaria occurs, but you don't get immunity to infections. You get immunity to disease, but not infection. You can get repeatedly infected over and over and over again, and you don't develop good immunity to that infection. And that's an immunology lecture, which I won't go into, but it's important for our story. So as I talked about this holoendemic malaria, what does that mean? Well, as we know here after our long winter, our mosquitoes are long since gone, and I keep on thinking that's a good reason for a long winter. But we know that when the summer comes back, the mosquitoes will come back and we'll have outbreaks of, say, mosquito-transmitted diseases like West Nile virus that's common here. So in parts of Africa that are actually higher elevation, you can have this unstable transmission of malaria just like there because it's transmitted by a mosquito. The mosquitoes come and go. And so you have outbreaks of malaria, but it's not year-round. However, in areas where it's stable transmission or holoendemic, the mosquitoes, it's on the equatorial plane. The weather's really nice. I like the weather, especially in March. And it's about in the 80 or so, and the mosquitoes live year-round. And if the mosquitoes live year-round, that means they can transmit the parasite year-round. And so the parasite is transmitted to these kids typically in an area where it's holoendemic 3 to 10 times per year that they get infected with malaria. So what I like to think about in an area where malaria is holoendemic, that it's considered a chronic infection of children in these regions. So they get infected over and over again. I kind of think about malaria, you get this systemic red blood cells and it's everywhere, right? So your blood cells are circulating, you get the parasite. So it's just this It's knocking on your body system. It's making your immune system work really, really hard. So it's not a good thing. So coming back to the story of Burkitt's lymphoma, that's a little segue into malaria. We know that Dennis Burkitt, he sees this map, and he's got these cases, and they're found in the areas where it's malaria. And he goes to England and presents this talk in 1961. Now I just was at a meeting. This slide note, seminar notice, was saved by Tony Epstein, who had gone to this talk, and I was just at the 50th anniversary of the discovery of Epstein-Barr virus, which I'll talk about in a moment, and he still had that seminar notice from 50 years earlier. And I always like to tell my students, this is why you should go to seminars, because you never know what you're going to hear. And what was at that seminar, and why he was so excited, was that Dennis Burkitt was describing this restriction of this cancer to a certain region. And at that time, we knew that cancers could be caused by viruses that infected mice and chickens and rats, but we didn't think there was a virus, or we were hoping, but they were looking, is there a virus that could cause cancer in humans? And this was the big question that got Tony Epstein so excited. So he set up a collaboration immediately with Dennis Burkitt. And Dennis Burkitt every Friday would ship up biopsy samples from Uganda up to England. And it took him 2 to 3 years. But eventually, what Tony Epstein saw in these tumor cell lines is a picture, electron micrograph, of a virus within those lymphoma cell lines. And it's a fascinating story what Tony Epstein, the work it took him to find that. So now he finds this virus. This is exciting. The virus is in this cancer cells. We found the virus that causes cancer. So then they look to see where the virus is, and guess what? It's everywhere, right? So this is a big like, oh, huh. So Epstein-Barr virus, or we call it EBV, is a virus that infects, I would say, probably everybody in this room except for maybe 1 or 2 of you. Greater than 95% of the population worldwide is infected with this virus. It sits there quietly, doesn't do anything, doesn't bother us. However, if you don't get infected till you're a teenager, and EBV, Epstein-Barr virus, is linked to causes infectious mononucleosis. It used to be called the kissing disease because it's transmitted through saliva. And usually you have a lot of saliva exchange early in life, and then there's sort of this gap there, and then the teenage years come in and saliva exchange begins again. It's also associated with a number of cancers, not only Burkitt's lymphoma, but cancer called nasopharyngeal carcinoma, which is very common in southern China, Hodgkin's lymphoma, a certain subtype of that, gastric cancer, and those are in immunocompetent people. It's also found with cancers in immunocompromised people as well. So it's listed by the International Agency for Research in Cancer as a class 1 carcinogen. So this is a virus we all have, but now we have to understand, how does this virus emerge to cause cancer? If we all have it and most of us control it, it comes out in a very small subset. So I want to just tell you, this is a question that's been bugging me for a long time, trying to figure this out. And I don't want to go too much into the life cycle in B cells, but it's important for us to understand this because I'm going to tell you a little bit about the work we're trying to do to get at that question. Here's the virus right there. It's in the saliva, and when it gets into the saliva, it gets into the B cells that are right in your tonsils. Your tonsils have lots of B cells in them. And it infects those cells, but what it does, which is different than most viruses, usually pretty much every virus that you get infected with will cause what we call a lytic replication. It'll divide and make more of itself. You get flu, a lot of those symptoms is because that virus is dividing and it's getting ready to go somewhere else. This virus comes in and goes quiet, and it becomes what we call a latent infection. So you eventually find it in your peripheral blood, in the B cells, and it's a quiet infection. It's just the viral genome is there. It's just biding its own time. And it's a very rare component in U.S. adults. I call it the healthy U.S. adult model. 1 in 100,000 B cells are positive for this virus. Very, very rare. Sometimes what can happen, we shed it in our saliva. It can go from this quiet stage to a replicating stage, and we shed it in the saliva again, which is how it can be transmitted, and it can infect more cells. So I was thinking about the life cycle of EBV and thinking about how could it be working with malaria. And there's a number of different models, but what we thought malaria might be doing is the infected red blood cell can actually come together with the B cell in the spleen, where it's a big problem with the clearance of the red blood cells during a malaria infection. And we know that the malaria actually has molecules on the surface of the infected red blood cell that can actually reactivate and make the virus go from latent to lytic. We've seen this in cell culture model systems. And we also know that it also has some ligands on it that could make that B cell divide. So we hypothesized that the malaria might be driving those B cells to make more B cells that are infected with the virus and basically increase what we call the viral load, or the number of latently infected B cells in the blood. So to do this, to answer these questions, we're actually working in Kenya. I like to point this in East Africa, and we work here on the coast of Lake Victoria. Lake Victoria is the second largest body of water. Lake Superior is the first. Sorry about Lake Ontario. I don't know where it quite fits in, but it's a very large Great Lake. And I put this map of Kenya relative to the East Coast because it gives you a sense of the scale of the country. It actually sort of takes in all of New York, part of Canada, pretty much the whole East Coast there. So it's a fairly large country in size. And this is just pictures from Kisumu where we work. I've had people say, oh, how are the lions at night? And I'm like, I have my gin and tonic looking at the sunset over Lake Victoria. So it's not so bad. And that's the Lake Victoria there, and didn't quite capture the sunset that night. And then it's your typical African town with, it's your basic about a 500,000 population in the city and maybe 1 million total. So actually not much difference in size than Albany. This is a clinic we work at, so we're just not too far out of town. Again, like Albany, you can get rural very quickly. This is a mother at the clinic weighing the child in one of our studies just to get a weight of the child. And this is our clinical officer, David Madem. And you can't see it quite well in this picture. He's got his hand on the child's head, and he's holding his hand there, and it's very sweet. In Kenya, we have this hospital, actually. There's no medical doctors assigned to the hospital, only nurses, actually. And David Madem is a clinical officer, which would be the equivalent of like 2 years post-high school training. And this is where most of the primary care comes from in Africa. This is just a couple pictures out in the clinic. This is a traditional hut. This is the road, and you can know the car is tilted that way because the car is tilted that way. Driving there is a little challenging. This is in the highlands of Kenya. They have the beautiful tea plantations, and this is just a market there. So you can be out in these beautiful rural areas fairly quickly, and we work in 2 different rural sites there. So what we did early on was we mapped out the cases of Burkitt's lymphoma because we wanted to see where the different cases were. And we have 2 sites that we work in up in the highlands of Kenya in the Nandi region. Those tea plantations, Kenya, they have the Great Rift Valley, so it's about 3,000 meters up, so I think it's about 8,000 feet elevation. The mosquitoes, because of the coolness up there, don't live year-round, so we have sporadic outbreaks of malaria. In Kisumu, down on the lake shore, you get malaria transmission, actually some of the highest in the world, year-round. So you have holoendemic malaria, and you have a high incidence of Burkitt lymphoma there. So we have this natural variation about 3 hours drive apart between areas that have a lot of malaria and a little malaria. And as I told you, EBV is found everywhere, so that's not a difference between the 2 sites. And so one of the first things we did was we just asked that question. Remember, I thought that maybe there would be more virus in the blood if there's more malaria. And the answer is in these 1 to 4-year-old kids, we did what's called a cross-sectional study. So we just went out to out into the community and drew blood samples. And actually, we didn't just do that. You actually have to go in. You have to get permission from after you get your IRB approval here, your ethical approval there. Then you meet with the village chief. Then you have a baraza and gather the community together and tell them what it is that you're trying to do. And then you draw your blood sample. And you can see this is the amount of virus in the blood in people from Nandi, the children 1 to 4-year-olds from Nandi versus the ones in Kisumu. So where there's more malaria, there's a lot more virus in the blood. The challenge with this, though, is that this is 1 to 4. It's a cross-section in time, so we don't know how did it get to be so high? So we decided to do another study. And in that study, we followed the infants in these 2 different sites again, longitudinally. So we followed them from 1 month of age up through 3 years of age to look at when they got infected with EBV, when they got infected with malaria, and what happened. And this is just pictures. This is my student who's now an assistant professor, Molo, in the laboratory. We work at the Kenya Medical Research Institute in Kisumu. It's a part of the Center for Global Health Research. There's the CDC is there. Walter Reed Army Institute of Research is there. A lot of other universities have laboratories there. So it's a very active environment to do research, and we have our laboratories there as well. Oh, so we enrolled a number of kids. We analyzed 150 kids. What we found, which isn't what I was looking for, which is why you always do science, is I wanted to see if they got malaria, how that would be working, and it turned out I couldn't do the malaria piece as well. But the kids were infected. This is kids in Kisumu. That's the one with lots of malaria. At 7.2 months of age, actually much earlier than the kids from Nandi, 8.5 months of age. But the striking thing is there's a number of children that were infected at less than 6 months of age. Now, the studies on EBV primary infection have all indicated that EBV isn't transmitted maybe 1, 2, 3 years of age. But no one's actually ever worked in a rural area, which is where we're working, and the hygiene is a lot different in those sites. But if you're looking at, in the region in Kisumu, you have 36% of these kids are infected before 6 months. That's a pretty striking number. And so the question is, if you're infected before 6 months, you really have to look at the mom, right? So you have to go back even earlier. So we decided to do another study. Oh, there's one more piece here. This is, the other thing that we wanted to do was, what does that mean if they're infected so early in life? So then we also measured their viral load, how much virus is in the blood over time. And what we found, and this is complicated statistical modeling, but if you were born in Kisumu, you had an early age of infection, or you had malaria positivity, all of those increased your risk for having an elevated viral load over time. What it means is that you're not controlling that infection very well. So you're getting infected with this virus early in life, the malaria, you're not controlling it. So you have more of these virally infected B cells. So as I said, we decided to do another study, and this time we decided to focus just on the area in Kisumu region where there's a lot of malaria, and focus in and look at the mothers at this clinic and go backwards in time. So what we did is we enrolled mothers at an antenatal clinic near Kisumu, the same one we've been working at actually. And we enrolled 175 mothers. We actually enrolled 200 mothers. 25 mothers were HIV positive on the first time diagnosis there. So I mean, that gives you a sense of the prevalence of HIV in this population. It's quite high. There's treatment for the mothers through a program through the PEPFAR. US government actually provides treatment for that. So they were not included in our study. The mothers were followed up through the antenatal visits. They come into the hospital. We actually pay for the mothers to come in for their ANC visits. And when I say we pay for them, we pay their transport reimbursement. And we time their ANC visits to market day so their husbands will let them come in to come and get the antenatal care. And then they can go to market and do their shopping. So this is a fairly complicated slide, and it's hard to read the numbers. But what we did is looked at EBV in the blood of the mothers then to see if malaria was affecting what was going on in the moms. And what we found is that the mothers that had malaria during pregnancy had more virus in their blood as well. So that the virus was being reactivated, or the malaria was affecting EBV in the mothers. And so I'm going to wrap this up in a sense and think about how do EBV and malaria interact to increase the risk for Burkitt's lymphoma. So this is my trying to put it all together model. And this is the thing that I keep on coming back to sort of as the key question. So this is my current model right now. What I think happens is during pregnancy, the mother gets infected with Plasmodium falciparum malaria. In these areas where malaria is endemic, this is fairly common to happen. That causes the mother now, she's infected with EBV just like everybody else is, but that virus reactivates and she starts shedding the virus. Data I didn't show you is that we can find and actually detect the virus in the breast milk. So we think that the mother's now transmitting the virus to the infant through breastfeeding practices. That's not actually been described before, but we have fairly good evidence to demonstrate that. What that means is when those kids are infected that early in life, that means they're going to have more of these B cells that are infected with the virus, right? And what happens then is these kids get repeated malaria infections, and now instead of having this many B cells infected with the virus, they have this many B cells infected with the virus. So it increases their viral load over time. And so I've been thinking a lot about what does that mean to have this more virally infected B cells? How does that increase their risk? Well, there's one other piece to the puzzle, and I'm not going to show you the data, but this Plasmodium falciparum, that's my little parasite right there, can induce something. In B cells, they have to respond to parasites and bacteria, and there's certain recognition molecules on those parasites. And Plasmodium falciparum can trigger an enzyme called activation-induced deaminase. Now, what that enzyme does is normally in a regular B cell, it makes that B cells make the antibodies different and change them around. But it can also been shown in mouse models to induce that c-Myc translocation. And remember, I told you that c-Myc translocation, that overexpression of that gene, is a characteristic of Burkitt's lymphoma. What we think is happening is that one of those latently infected B cells is actually getting induced it causes c-Myc translocation, and that ultimately leads to the malignancy. So we've got some data where we're trying to hone in on this, and I won't show you that tonight because I'm trying to keep it a fairly broad talk about that. But I think our model is actually convincing us sort of where we need to focus. What I'd like to do in our next study, if I'm so bold, is I'd like to actually, what I'm proposing is that we should vaccinate the mothers against EBV. And what that would do is boost the mother's antibody titers against the virus so that then she keeps the level of virus that she's shedding down and delays that infection till a later age, and I think that would reduce the risk for these children getting Burkitt's lymphoma. The other approach, and being successfully used in Africa, obviously, is the introduction of bed nets. There's work on vaccines, but that's still a ways off. So there's ways to reduce malaria, but I think really I'd like to see if I could, sort of my career ender, if I could say it, is to actually work on the vaccine for EBV. So the last part I want to talk to you about, and those are the stories we've been working on in the projects in Kenya, is thinking about building cancer research capacity. Because this is a really key problem. I don't want to be going to Kenya, and I love going to Kenya, but at some point, it's got to be the next generation, and it's not going to be the generation, well, maybe there'll be generation here, but I want it to be my Kenyan students. So we've been fortunate to be funded through a grant through the National Cancer Institute. NCI is very worried about the AIDS-associated malignancies. If the major cause of death is HIV/AIDS, AIDS-related cancers are also very common. So I've been working with Scott Remick, who's an oncologist, Leona Ayers, who's a pathologist, and myself, and then Walter Mwanda in Nairobi. And why I like to talk about this project is it's a nice coming together of sort of different expertises and background to tackle an important problem. And what we've done is think about it with clinical trials as sort of the center focus. We had three aims of this grant. I'm the virology side over here. Leona's the pathology side, and ultimately trying to bring in clinical trials for cancer treatment. So we want to strengthen diagnostic and research pathology capacity. We've done this, and again, NCI has provided funding, so we've improved the pathology laboratory from when I first started there, and I showed you those slides with the little lots of jars. And now we can do tissue microarray and immunohistochemistry, and we have a pathology video conference capacity, so we can look at slides in Kenya and look at them in West Virginia, Syracuse, or Ohio. I've been working on the capacity for virology laboratories, so we can do actually now, when we measure EBV viral load, we do by assay that we can do in Kenya with a quantitative PCR machine that we have there. We have a Luminex machine. I have a BSL-2 lab. And I'm going to talk a little bit about tumor registration in a moment. And then lastly, the clinical trials capacity through West Virginia. We brought people over to West Virginia University, nursing, pharmacology, so we have the capacity to actually test in country. And one of the things to think about is that it might be good to do clinical trials over here, but we have a different population base over there. So if there are new therapies, we really want to be able to test them out in the country where they're going to be used. This is a picture of our group. And what I'd like to, this is my student Sydney, who's getting his Ph.D. with me. It should be done in sort of the immunology aspect of the project I was telling you about. And career, she can find the time, we'll get her Ph.D. with me in cancer registry work. This is Ibrahim Daoud, who's actually here right now finishing up his Ph.D. and doing training at National Cancer Institute, and we'll be going back, I hope, to run a project we're going to do in Nairobi. Odata Sumba is one of our, he helped lead our project back in the late 90s, came and got his PhD in his late 40s, and now a senior cancer researcher and very important and really developing a lot of work with some of the groups at NCI. And Amolo Aceto, also my student, who got his master's and PhD with me in postdoc and now is an assistant professor. So when I started in Kenya, there was actually very little training. There's a lot in cancer at all. And I feel very proud that we've got a number of students that talk about that. They're getting students that want to talk about EBV and KSHV and cancer. And so I think, you know, it's a small seed to begin with, but it's definitely a seed that's growing. The other piece is the cancer registry. And as I told you, one of the challenges in Africa is understanding the burden of disease. Kenya Medical Research Institute started a cancer registry in 2001. This is mandated by the Ministry of Health, except all they gave them was a small office and one staff person, so not very much. That's some of those government mandates we all love so much. But it's been expanded in 2010 with the East African Cancer Registry. They're now working with Malawi, Ethiopia, Rwanda, Zimbabwe, Uganda, and Tanzania. And with our training grant, we've actually were able to train more cancer registrars and are now, in fact, beginning to do cancer registry in Kisumu as well through training through this group. And now there's 19 members of this African network being sponsored. This is Ann Carrera and Nathan Matusi, who trained with us. Through our training grant, we actually were able to buy 2 computers. They had I think they were using a 2001 computer prior. So it doesn't take much to build capacity when you think about it. We're not talking about a huge investment, but a steady and regular investment is actually key. And Ann actually just published a paper in PLOS 1 trying to look at strength of evidence to figure out about HIV malignancies, since that's what the training grant is about. And one of the problems is that when you look at cancer registry, if you're abstracting data, there's no diagnosis of somebody has HIV positive. HIV still has a stigma. It's hard to figure out. So what she did is to look at clinical evidence to see if she could find evidence of what types of cancers are there. So people might say they're on antiretroviral therapy or they have low CD4 count or other indicators, but rarely do you find somebody ordering an HIV test as well. And this just shows you, using this data that she's collected through the registry, we get a lot of squamous cell carcinoma, non-Hodgkin's lymphoma, cervical cancer, but Kaposi sarcoma is quite high. Kaposi sarcoma is also linked to a viral infection. Cervical cancer is linked to human papillomavirus. This is a viral infection. Not clear about that one. So there's a lot of work to be done, but I think that we've made a good start with this. So when I think about the challenges faced working in Africa and cancer research, as I said, there's a number of challenges that are there, but I feel like we've at least in Kenya in a small way, are making some inroads into improving those challenges so that if I talk to you in 10 years from now, they won't be there. But I also think about why do we do cancer research in Africa? I had somebody tell me, I was talking to somebody who said, well, do you get good wine there? And I said, well, Gato Negro. So you don't get good wine when you go there. It's not a high-end kind of place to go. But we think about from the work at Dennis Burkitt, studying cancer in Africa actually opened the door in our understanding. Guy Dete called it the Rosetta Stone of human malignancies. So here's some Irish surgeon off in Kampala in the 50s. And out of this was the first cancer shown to have a human oncogene. We now know most cancers all have oncogenic translocations or deregulation. It's the first cancer shown to be treated only with chemotherapy. Dennis Burkitt couldn't use radiotherapy, that wasn't available, so he tried chemotherapy alone, and in this particular case it worked. That changed the paradigm for cancer treatment around the world so that people switched over for many cancers with chemotherapy rather than just radiotherapy. It was the first cancer shown to have, human cancer, to have a viral association with Epstein-Barr virus. And actually, now we know about 20% of cancers worldwide are caused by an infection, right? So cervical cancer is linked to human papillomavirus, liver cancer, hepatitis B and hepatitis C, bladder cancer, schistosomiasis, gastric cancer, Epstein-Barr ranks up in a lot of different ones, Burkitt's lymphoma, and so on. So there's a number of cancers that are linked to human viruses or parasites. And it was through the study of Dennis Burkitt that really made that. But I'm going to stop here. And I think the other reason, as I said, when we come face to face with cancer, we also have to think about the children. In these pictures, my husband went with me to Kenya, and those are his sunglasses. The cost of the sunglasses is the same cost of chemotherapy. $150. But the kids were having a great time because they had the glasses on and looking at their picture, and I just really like this series of pictures. And I think that's the other thing we always have to remind ourselves of why we do the work there. So I'm just going to stop there. The work is done with a group at Kemry. This is my staff and team there. It takes a lot of work to do those field studies. This isn't quite everybody, but I always take everybody out to lunch for fish on the lake when I go. And then I also have a lab here in Upstate, and thank them as well. So I'll stop there and take any questions. Bill?
Quick comment and a question. I was noticing you said the life expectancy is about age 46.
Mm-hmm.
That's exactly the same it was in 18th century colonial America. That gives you some perspective going forward. My question is, you talked about vaccinating to reduce viral load.
Right.
How effective is that, and over what period of time would you think that viral load could be kept down? And the second part of it is, as with HIV/AIDS, where we give a cocktail now to reduce viral load, could this work with ABV as well?
So,
Dr. Rochford, if you
yeah.
wouldn't mind just repeat the question briefly for the folks who are in the room.
all right. The question was, is how would the vaccination work in terms of reducing viral load? What I'm proposing is actually vaccinating the mothers to induce their antibody titers so they'll be shedding less virus, so they'll transmit the virus at a later age to the kids. There are some proposals to have therapeutic vaccinations so that you could target and help the immune system fight the virus, and that might be another model system. And I think both of those approaches are worth investigating. The second question was,
I think you answered both.
all right. In the back?
Is there any crosstalk between parasite and the virus at genetic level?
Is there any crosstalk between the virus and the parasite at the genetic level? Well, the parasite is infecting red blood cells, which are enucleated, right? So we don't see much of that. We have the parasite. Just the parasite DNA is signaling the B cells, but beyond that, we don't think there's any other interaction.
There's no interaction between the viral genome and the parasite genome.
No, not that we know of. Might be worth looking, though.
Is there any protective effect of S-hemoglobin in patient
Right.
population?
In the sickle cell. So in the African, the question is there a protective effect of S-hemoglobin? In Kenya, it's about 18% of a sickle cell trait. You don't get sicklers in Kenya usually because they don't survive, because there's little treatment. So there was just a study done by a colleague of mine, Ann Mormon, who showed that there was actually no protective effect, a fairly larger study. There were some earlier ones suggesting there might be. But remember, with sickle cell, it doesn't prevent infection, it just prevents disease. So you still, the parasite, you still get infected with malaria, just the parasite doesn't replicate so well.
Right.
So.
Do the tumors on the children, do they go away from the chemo?
They do, actually, and I should have brought a picture because I have this great, within 2 weeks, they'll actually almost melt away. And the faces that are so distorted with the bones out of place, they go right back in place. Within just the 2-week window. Sometimes the mothers will take their kids home early, which is a problem because they see the tumor go away so quick, but it does.
What is the recurrence rate?
The question is, what is the recurrence rate of Burkitt's lymphoma? And that's a great question. The problem is we don't know the answer. So in this country, if you're treated with Burkitt's lymphoma, it's about a 90% cure rate. And I'm not sure about the recurrence here, but in Kenya, trying to figure out what the recurrence is very hard because you have to track these children home to very rural areas, so we don't know that. The cure rate, though, 6-month cure rate is about 50%, so much lower, and there's relapse, about 10% is estimated from a study done in Malawi.
Is the occurrence rate in this country when you have Burkitt's, is it from EBV or because we don't have much malaria?
Right. So the question is, in this country, when we have Burkitt's lymphoma, what it's due to, and we don't know. It's what I sort of say is in the incident rate of recurrence in our hospital in Syracuse, we'll have 1 case in a year, which is a large county hospital if you think about it. In Kisumu, we have 2 cases a week. In this country, so it's very rare, it's sporadic. So we don't know, it's not associated with EBV typically. So we don't know what drove that. I always say it's convergent evolution. That c-Myc translocation is found in all those lymphomas, but in Africa, the malaria, I think, is driving it, and something else must be driving it here. But it's so rare that nobody's studied that.
I have a question regarding the state of funding
Mm-hmm.
for your particular research in particular,
Right.
as well as cancer research in general in Africa, and the challenges now as well as what you foresee and the outcome from it.
So the question is about the state of funding now and in the future. So fortunately, right now, the head of National Cancer Institute, Harold Varmus, has made global health cancer research a priority for him. Now, has he translated that to more grant dollars for me? I'll tell you next year. But there is a big push by NCI to do more work on the larger global level. But NIH funding all across the board has been cut. My grant kept on getting cut, 10%, 10%, 10%. I was fortunate that I found other avenues to make up the difference, but it's challenging.
What about from private foundations?
The Gates Foundation doesn't deal with, they'll do cervical, they do HPV work, but Gates Foundation doesn't do cancer work at all. They leave that alone. There's been an organization that raised funding in University of Washington area where they have some of the people who made money off the Microsoft. I started a small nonprofit years ago trying to do that, and it's a tough thing to do. Yes?
Are there any techniques using remote medicine or remote diagnostics that could sort of decouple the point of care, or at least from a diagnostic perspective, for early stage detection and its diagnosis on a remote or virtual basis?
Right. So the question is about early stage diagnosis or early point of care diagnostic for detection of Burkitt's lymphoma. But one of the big problems is actually what we thought about is getting the clinical officers, who are really the ones that are out in the rural areas, being the ones to get more cancer awareness. And I think once we get that piece in place, there's a lot of groups doing telepathology right now, but it requires a skilled surgeon to do the fine needle aspirate. So you actually still need some high-level training to do those biopsies. All right.
Rosemary, I'd like to thank you very much. There's a tradition with the Hogarty Lectures, which Dan knows very well. We always present a plaque for these events, which is actually an etching of the invitation that many of you received. So Dan, if you could come up here. I hope these are on your wall somewhere.
Yes, we have. We have room on the wall for this one.
Okay. And this one's for you.
Oh, well, thank you very
One
much.
of the things you guys won't know probably is that I grew up next door to Dennisburg in Uganda. So this is showing the Lago Hospital to me. It's just like going home, and it was a really tremendous thought. Absolutely.
Thank you. And thank you very much. Thank you.
Thank you.